Hello everyone, I'm Jonas Korlach, Chief Scientific Officer at PacBio, and I want to welcome everyone to the PacBio webinar series, with today's topic featuring our new Sequel II system. It's my pleasure today to introduce the speakers who will present on their experiences with our new Sequel II system in the following order. Kiran Garimella, a Senior Computational Scientist at the Broad Institute of MIT and Harvard University, followed by Luke Tallon, Scientific Director of the Genomics Resource Center at University of Maryland School of Medicine, and Shane McCarthy, Senior Research Associate at the University of Cambridge. We have a lot of great material to cover today, so time permitting, the presentation portion of the webinar will be followed by a questions and answers session. You are welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee dashboard. We will be recording this webinar and making it available for download in the next few days. Please keep an eye out for a follow-up email with a link to the recording. Before we get started with the three presentations, I wanted to very briefly offer the context and scope of today's webinar. We wanted to all say hello, but we will now turn off our webcams so that you can focus on the slides. And so as for context, in April of this year, we launched a new Sequel II system, which compared to the Sequel system has an 8 times higher feature number, and that results in an 8 times increase in yield, translating to a reduced project time, lower cost, while maintaining the equivalent performance of what you're all used to with regard to the quality of the PacBio data of providing very long reads, high consensus accuracy, minimal sequence context bias, the unique ability to generate long and accurate HiFi reads, and the ability to detect methylation signatures. And in the 3 months prior to the official launch, we had selected 5 early access sites, 3 of them you will hear about today, and we thought it would be very informative for you to hear about their experiences during that early access time period. And we, of course, chose the early access program to make sure that the system performs as it does here at our facilities at external laboratories and institutions. And so with that, I would like to pass it off to our first speaker, Kiran Garimella, and we'll switch over to his presentation. And thank you all for attending, and we'll now start with the 3 presentations.
Great. All right. Thank you very much. My name is Kiran Garimella. I'm a senior computational scientist here at the Broad Institute, and I'm grateful for this opportunity to talk about the Broad's recent installation of the PacBio Sequel II and some of the lessons that we've learned from our initial 30 or 40 runs of the instrument. Before I get started, there are a number of people that I am presenting on behalf of who have done a lot of work behind the scenes. In particular, Maura Costello, who's been doing 100% of the library construction and physical sequencing efforts at the Broad. And Roberto, who's been my contact at PacBio, who's given a tremendous amount of useful feedback in getting our instrument and data processing workflows up and running. So primarily, the Broad has been interested in using the new Sequel II instrument to generate the CCS, or what's now called the HiFi reads. And if you're unfamiliar with this data type, briefly, what occurs is a long double-stranded DNA fragment is isolated from the genome. These circular adapters, these bell-shaped adapters, are ligated to either end, and then a polymerase is affixed to the structure, which begins sequencing in a circle, displacing previously sequenced bases as it goes along, and resulting in these this pattern of forward and reverse subreads with the circular adapter interspersed. The adapters are easily computationally removed, and then all of the subreads are collapsed into a single high accuracy consensus sequence. As part of our initial evaluation of the instrument, PacBio has given us 32 smart cells and reagents for use on various projects of interest to lab members at the Broad. We divided those samples up into basically two bins. On the left-hand side are data sets that largely overlap with the HCSV consortium efforts. So a lot of trios from the NHGRI and HapMap sample collections at Coriell. All of that data was chosen because we intend to release it publicly through HCSV to the world. And then on the right-hand side are a number of clinical samples. These reflect trios where a cryptic variant, we hope, underlies some interesting Mendelian disease. It reflects tumor normal cell line pairs that we're hoping to use the instrument to elucidate some of the more complex SVs that are occurring in those samples. And there are some repeat expansion disorders that our clinical partners have been interested in examining in greater detail. Oops, what's happening here? Okay, well, hopefully you can still see the slide this way. So on average, per smart cell, we're seeing around 300 gigabases of data per smart cell. As the instrument does a number of circular passes around these reads, we're seeing around 10 passes per read. We have run PacBio's circular consensus error correction tool on the data, and on the left-hand side of this page, I'm showing data before error correction, after error correction, and then some of the NIST Platinum Genomes data for comparison. So before error correction, we're seeing error rates that are around 10%, Q10. After error correction, we see error rates drop by approximately an order of magnitude, somewhere between the 23 and the Q23 and Q25 mark, depending on whether you're measuring for mismatch errors, insertions, or deletions. For comparison, the recent data made available through the Platinum Genomes project, which are Illumina data sets, come in at an average of Q24, Q25. And obviously the indel error mode in Illumina data is much, much lower than it is for PacBio data. But still, after correction, we're seeing comparable error rates between the two data sets, which is pretty remarkable. Of the remaining errors that we see, 94% of them are simply one base insertion and deletion errors, which are fairly easy to deal with algorithmically. One of the big challenges that we had in getting our pipeline up and running is because the Broad is a production center, it's become increasingly inefficient for us to run the computation on local compute, as we are constantly on the verge of filling up hard drives or simply not having enough CPUs to run some of the more algorithmically intensive algorithms that we'd like to run. We've solved that problem for Illumina data by migrating all of the compute to Google Cloud, and we've done exactly the same for the PacBio data as well. So 100% of the workflow that we've implemented is purely on Google Cloud Compute. And that requires a little bit of care in terms of processing the data, making sure that all of the data that should be corrected together is corrected together. Those tools are being released as part of the GTK. They're available now at the link below, and eventually these will migrate to the master branch. Similarly, with the workflow description that we use in production for all of this data, that is being released publicly as well at the link, the GitHub link below. Briefly, the way that our processing works is we take the uncorrected BAM file that's emitted by the PacBio instrument, and that is the final processing that is done on premises at the Broad. After the instrument writes it out to a local disk, it immediately gets uploaded to Google Cloud Storage, where we split it up into around 300 to 400 different shards. Each shard is corrected using 4 threads, so we're running around 1,200 to 1,600 fold parallel in the cloud. And similarly with the variant calling, we're throwing basically every variant caller that we can find, also in parallelized fashion, so that we can make calls with the GATK, deep variant, PacBio's own PBSV tool, Sniffles, Svim, I think, was added to the pipeline yesterday. As a result, this pipeline runs end to end from uncorrected data to variant calls in somewhere between 1 to 2 days. It depends on whether you opt for Google Cloud's ability to give you high priority compute, in which you don't share the nodes with anyone, or low priority compute where other users of the cloud may preempt your jobs, meaning they take a little bit longer, but the nodes themselves are 5 times or 1/5 the cost of the high priority nodes. One of the really interesting things that we're looking at with these data sets is their ability to establish a ground truth for classes of variation that we've historically not had ground truth data for, particularly SVs that are greater than 50 bases long. And in this screenshot, I've put together what I think is a nice example of the challenges that we face with short read sequencing and why the Broad is so excited about long read sequencing. So in the top panel, I have some data from NA12878 that was from a recent Illumina HiSeq run. And as you can see, there seem to be 3 things going on in this panel. First, there's a variant on the 5' side and another heterozygous variant on the 3' side. And then in the middle is some cryptic event that was not called by any of our short read callers. There are a lot of reads that are mismapped here. There's a lot of sequencing error rate noise. There seems to be a dip in coverage. So visually, it's apparent that perhaps something is going on, but it's very difficult for the short read based structural variant caller to make a definitive call as to what the nature of this event is. Plus, the phase between the 5' heterozygous site and the 3' heterozygous site is not at all elucidated from the short reads, which just don't span this nearly 750 base region. In comparison, we have 2 replicates of the NA12878 data set on long reads. And then the mother and father for that child as well, all sequenced with PacBio CCS reads. And what becomes clear is that this variant is very clearly in the, I think this is the mother, absent in the father, and then present in both replicates of the child. And furthermore, these variants, which we could not phase before, now visually their phase is very clear that they are both in phase with this nearly 300 base deletion in the child that's been inherited from the mom. Here's another example. This is a 32 kilobase locus, and the region that I've highlighted in red harbors a fairly large inversion, almost 1,200 bases long. But because that event is so large, you would imagine the signal in the Illumina data is not quite clear, Whereas in the PacBio data, we have a clear as day signal with a lot of reads that seem to map correctly right up until the point where they hit the breakpoints for the inversion event, and then it appears that all of the bases are suddenly mismapped to the reference. And by just looking down the column, you can tell what the inversion sequence is. This is another event that we're particularly excited about because it's not clear how you would call this event with the Illumina data. The PacBio data makes it very clear as to the nature of this event. Other things that we're interested in are looking at regions of the genome with high genomic diversity. For example, the HLA on chromosome 6. On the top panel, once again, I have the Illumina data for this region for one of our samples, and you can see that there's a tremendous amount of coverage variability across the gene body of HLA-B versus in the PacBio data, where we see very consistent coverage throughout the entire locus. And one of the things that we're doing right now is developing an HLA caller that will run on the long read data for all of the samples that we run through our production pipeline. So to wrap up very quickly, I think we're very impressed with the PacBio the Sequel II data. The error correction does seem to work very well, reduces the error rate by more than an order of magnitude, bringing it in line with the data quality that we expect to see from sort of typical Illumina runs. We're getting around 10x coverage of corrected data per SMRT Cell with quite uniform coverage over the diverse regions of the genome. All of the pipelines that we're producing are available publicly in the GitHub link that I've mentioned below. Like I said, depending on whether you opt for low priority or high priority compute, this runs for somewhere between 1 and 2 days for a total of $30 to $150 per sample. This will eventually be featured as part of the online pipeline tools that the Broad has been releasing, called, it used to be called Firecloud, now it's called Terra. And finally, the particular exciting thing for us is the long read's ability to elucidate, or rather enable the discovery of SVs that have been previously undetectable for us with short read data. And this is an ongoing area of development for us. When we have finished constructing the truth data sets for the HGSV trios, that's going to be released publicly so that anyone who's working on variant callers will have some truth data set to work with. And with that, I will hand it back to our next speaker. And I'll look forward to your questions at the end of the talk.
Great, thank you very much, Kiran. And we'll hold the questions and answers for at the end of all 3 presentations. So I will hand it off to our next speaker, Luke Tallon.
All right, thank you very much. And thanks everyone for attending. And I want to thank PacBio for inviting me to share our Sequel II early access results with all of you. I'll start by saying that there are a lot of data on some of my slides, and I'm going to really focus on high-level summaries here for my short talk. But I included those data for those of you who might want to go back to the recording of the webinar and take a look in a little bit more detail. So our early access started in late January on the 23rd when our instrument arrived. It was installed the next day, and we put up our first run of an E. coli test library the following Monday, which yielded an impressive 109 gigabases of total data. And a couple of days after that, we were running our own libraries. So the first thing we ran was an insect long read library, which yielded 94 gigabases of data. So it was a very good start for us. So we ran 32 total SMRT Cells during early access, 2 of which failed. And they were broken down across a pretty broad range of applications. So we ran 14 SMRT Cells of CLR, or continuous long read libraries, on humans, plants, and insect samples. 6 of the HiFi style CCS human SMRT Cells, and 12 other SMRT Cells that were a mix of microbial applications, whole genomes, amplicon profiling, and metagenome sequencing. And those will be the focus of most of my talk today. But before we get to that, here's just a summary of some of our long read runs. So you can see that they range from as low as 40 gigabases on what was a particularly difficult plant sample to as high as 132 gigabases of yield on a human sample, with an average of over 90 gigabases of total data. And on the read length side, we had average polymerase N50s almost 40 kb and average subreads of almost 35 kb. So very nice long reads from long libraries. As a way of direct comparison, we ran a couple of different libraries on both our Sequel and Sequel II systems. So I'm showing one example here. This is a plant genome that we sequenced with a long library that was about 85 kb in insert length with a 30 kb size selection. So you can see in the top panel that we were averaging just over 10 gigabases of data on the Sequel system. And when we tried this exact same library on Sequel II, we yielded over 115 gigabases. So over a 10-fold increase in total yield, with about an 18% increase in the raw polymerase read length and a 20% increase in our subread N50s. So pretty impressive increases there. On the human HiFi side, we only ran 6 cells, and they averaged about 260 gigabases. With nice long reads. After we ran CCS, where we used a minimum of 3 passes around that circle that Kiran was describing with a Q20 cutoff, we had a mean yield of about 16 gigabases of total data per SMRT Cell, with a mean quality score of just over 32. Our other runs, which, as I mentioned, were primarily microbial, varied quite a bit, as you might imagine, given the different sample types. So you can see those here. We had some bacterial long reads, we had bacterial multiplexing, 16S amplicons, and metagenome runs. So these runs, although they appear more varied, actually met our expectations in terms of what we thought we would get from given application types and library types. And so these are the runs that I really want to focus on here today. So I'm going to talk about 3 different experiments we did. The first is bacterial whole genome multiplexing. So for this, we selected 16 bacterial genomes, 15 of which we have complete, curated, polished reference sequences that we generated from non-multiplex libraries on either RS2 or Sequel. And the pool totals about 80 megabases. We chose the genomes across a range of genome size from about 2 to 7 megabases, across a range of GC content, repeat content, as well as plasmid size and plasmid content. Each of these samples were sheared with a 10 kb insert size target, and we applied a 5 kb size selection. So here you can see the overlap of all 16 samples. So pretty reasonable and tight distribution for the pooled library. This library was sequenced on a single SMRT Cell that yielded about 215 gigabases of total data. After running it through CCS, again with the same 3-pass Q20 cutoff, we yielded about 1.4 million CCS reads and about 12 gigabases of total CCS data. So we then demultiplexed these data both at the raw subread level and at the CCS read level. On the right, you can see the distribution of post-demultiplex read count for each of the 16 samples. So there is some variability there, and we in particular had 3 samples that were a little underrepresented in this pool, both at both levels. But overall, the demultiplexing was very high. So you can see on the left that at the subread level, we got almost 95% of the reads accurately demultiplexed. And at the CCS level, almost 99% of the reads were accurately demultiplexed. So each of these data sets were assembled and compared to our reference sequence. So we assembled the CCS reads in their entirety for each sample, And then we assembled a randomly subsampled set of about 100,000 raw subreads. And we chose that number because it mimicked about the amount of data, both at read count and read length, that we'd expect from a single non-multiplexed RS2 SMRT cell, which is sort of our gold standard for a single bacterial genome on PacBio. And as you can see, with the exception of one sample, which turns out was a contaminated co-culture that we discovered and contains multiple, the rest of the assemblies had a maximum contig count of 10. Most of them are considerably fewer in contig count than that. All of the assemblies achieved essentially complete genome coverage. And most of the assemblies had comparable contig N50s to the reference sequence. So of these assemblies, 3/4 of the CCS assemblies, 12 out of the 16, were able to assemble the chromosome of the bacteria into a single contig, and 9 of the 16 assemblies at the subread level achieved the same. So then we started to ask the question, well, that was 16-plex. How deep can we go? And we're still exploring that, but one first pass at looking at that was that we decided to further subsample 3 of the genomes on this pool and take a look at what the assemblies would look like at 75,000 subreads and 50,000 subreads, respectively. So you can see on the upper right what the coverage profile of each of those samples looks like in terms of raw coverage of the genome, total contig count, and contig N50 on the middle and lower right-hand side. So all of these assemblies achieved over 99.9% completeness in terms of genome content. For 2 of the 3, the Yersinia and the E. coli sample, all of the assemblies were able to complete the chromosome in a single contig. For the Bacillus sample, only the CCS and the higher subread assembly were able to do that. But all in all, it's telling us that we can certainly multiplex at a much deeper level than this, and we're starting to explore, based on genome content and goals of project, how deep we can go. So moving on now to the second application I want to tell you about, which is full-length 16S amplicon sequencing. So many of you may know that the 16S gene is used in the metagenome and microbiome world for taxonomic profiling. The vast majority of 16S profiling uses short amplicons, usually a few hundred bases long, targeting 1 or more of the variable regions within the 16S gene. But those very relatively low overlap. So the paper I'm showing on the left demonstrates that if you take the same microbiome sample and you target it with amplicons looking at different variable regions, 1 or 2 of them, the chances of a read from the same organism within that community being assigned to the same OTU or operational taxonomic unit by standard microbiome software packages is at best 50 to 60% and often lower than that. And on the right is a paper showing that if you look at what's called the V4 region of the 16S gene, which is perhaps the most commonly used variable region for a lot of microbiome projects for short amplicon sequencing, that the taxonomic accuracy of those data are less than 50% at the genus level. And so there's a lot of reasons to do full-length 16S sequencing. And with the Sequel II system, we wanted to explore what those data looked like and can we do it in a cost-effective way. So we took 2 sets of 96-plex amplicon libraries. These are primarily infant gut samples, 180 of those, and then supplemented with a couple of control samples from a vaginal microbiome project we have, a couple of mock community samples, and our standard negative and positive controls. And each of these 96-plex libraries was sequenced on a single SMRT cell, so I'm showing one example here. On these cells, we were a little low in our loading, so we only were able to achieve about 30% loading on the SMRT cell, whereas we typically target 50, 60%. So in subsequent runs, we've done a better job with that. So we had a little bit less data here than we had hoped, but overall, we yielded about 112 gigabases of total data from about 2.5 million reads. After running that through circular consensus algorithm, we had about 1.2 million full-length reads that made it through CCS. We then looked at the quality of those reads, and as we expected, the quality was very high. So on the left, you can see a plot of average quality for one sample, one data set out of this set across the length of the read. And you can see that it is not position dependent, it remains high, and it maxes out at about Q80. And we compared that to the same sample run on Illumina, where you can see the expected 3' degradation in quality. And I should also note that even though these plots look the same height on the slide, the y-axis is different. The Illumina data maxes out at about Q40 and of course degrades quite a bit at the 3' end. So we then demultiplexed the data, and you can see the results of one example here. There was a little bit more variability in the pooling here than we are used to seeing and than we expected. So we had about 25% of the samples with relatively low coverage, fewer than 2,000 reads. This is about twice as variable as a typical 96-plex run on Illumina for us for 16S profiling. So we're working on ways to do a better job balancing the pool. But overall, we had about 12,000 CCS reads per sample. And all of these samples were then pushed through our standard analysis pipeline. So we do primer trimming, some quality score filtering and length filtering, and then they get run through a pretty standard 16S taxonomic analysis pipeline that we have here at the Institute. So following all of that, we yielded about 7,000 reads per sample. After we've gone through CSCS and all of our sort of downstream quality trimming, we assume that every variant in a sequence, or every sequence nucleotide change between one sequence and the next, is real and is not a result of sequencing error. And so we assign that as a separate amplicon sequence variant, or ASV. So we found on average about 17 ASVs per sample and a total of almost 1,400 different ASVs in the data set. Of those, over 1,200 of them, or almost 88% of them, were assigned at the species level. And the rest are at the genus level and above, although we think for the most part, those are due to a lack of full-length 16S reference sequences for those particular species in our databases. So we're working to add those to the database. But what you can see, both the plot on the left here and the numbers on the table on the right, is that we have a nice tight distribution of read length across different species in our data set. And for a number of these species, we're able to find dozens or even over 100 different ASVs that would lead us to sort of a subspecies level taxonomic assignment. So as I mentioned, as part of these runs, we included a couple of mock community samples. We used the Zymo Biomics community standard. And the theoretical profile of that mock community is the plot in the center of the slide here. The two PacBio runs we did on the Sequel II are on the left side, and two runs of the same mock community run on Illumina are shown on the right. And what you can see is that the PacBio runs, especially the second run we did, do a much better job of reconstituting the community profile that we would expect from this mock community. The first PacBio run we did, this was one of the low coverage samples, so we have fewer reads here, and we're a little high on one of the samples here and a little low on another. But both of these runs do a better job of representing the mock community than either of our Illumina runs here. And then finally, as I mentioned, we found a lot of ASVs or sequence variants that are at the subspecies level. So here's a figure that demonstrates that. So each shape here is one sample and one sequence variant that we found across all of the data set. And so as you can see, at a lot of these different species levels, there are many, many subspecies sequence variants that we're able to look at. So what we learned is that, as expected, full-length 16S sequencing is much more accurate on a taxonomic level than the shorter amplicons we use on Illumina, and that running them on Sequel II certainly gives us very highly accurate subspecies profiling. So the final application I want to talk to you about really quickly is doing metagenome shotgun sequencing. So following up on some of the 16S work. So we selected 5 samples for this initial test, and the questions we were trying to address were, can we capture full genome assemblies from these samples, which are from our vaginal microbiome project, with pretty limited starting material? In particular, we were looking to see if we could sequence and assemble an organism that's called the bacterial vaginosis-associated bacteria 1, or BVAB1, which is associated with negative outcomes in the female reproductive tract, and for which there's no fully complete reference genome sequence at this time. So of the 5 samples, 4 of them were vaginal metagenome samples with varying levels of expected BVAB1 content based on 16S sequencing, which you can see in the bar graph there. And the 5th sample was a cultured sample that was lactobacillus dominated, and for which we were trying to see if we could get strain level genome assemblies. So we had, as I said, limited starting material here. So we didn't have enough to do any shearing on the samples. We did very limited size selection. You can see from the overlay plot here that they were a little bit varied in terms of their fragment length and their overall content in the pooled library. We sequenced that combined pool on a single SMRT cell, yielded 191 gigabases, from which we got a little over 1 million CCS reads, totaling about 10 gigabases. The demultiplex results are on the bottom left here. You can see that they're a little bit variable, although part of that is explained by some of the lower abundant samples in the pool. We then took all of these data at the CCS level and did human read removal. So you can see that removed the vast majority of the reads from most of the samples. This sample here, if you can see my cursor, had very little human content, which is quite curious, and we're not quite sure why that happened. So we're looking at that, since it should have had a lot. But what remained was a relatively small set of data for each of these samples that we ran through some assemblies with Canoe and yielded relatively low contig coverage and N50s, as you might expect from such a limited data set. But what impressed us is that even though we had low coverage and we had low numbers of reads after all the human read filtering, we were still able to assemble complete genomes for 2 different Gardnerella samples. We were able to assemble the BVAB1 genome from one of the samples in 5 contigs from fewer than 200 CCS reads. And we were able to get a single contig genome for the lactose sample as well. Given the limited amount of data that we were able to achieve from these samples, we got pretty impressive results, and we're continuing to work at the subread level on these assemblies to see if we can improve them. So none of this work would have been possible without a whole bunch of people. So I want to thank the PacBio team that supported us during the early access program. I want to thank my team at the Genomics Resource Center here. In particular, I want to highlight Ellie Zhao, who did all of the library prep and sequencing for all of the samples. And I want to highlight Jacques Ravel's lab for the 16S and metagenome work, in particular Bing Ma, who worked on the 16S samples, and Joanna Holm, who did the metagenomes. So with that, I'll end. Thank you for listening, and I'll look forward to your questions after the webinar.
Thank you very much, Luke. And we'll pass it off to Shane McCarthy, our 3rd speaker.
Hi everyone. Yeah, thanks for joining us, and thanks PacBio for inviting me to talk about this. So I'm Shane McCarthy. I'm at the University of Cambridge with Richard Durbin's team there, but I work quite closely with the Sanger Institute, and I've been looking at the early data from the Sequel II system that we have up and running here. So from just a sort of, I'm coming from a slightly different background maybe to the other speakers, in that I'm currently involved in a lot of tree of life sequencing projects. So one project being the Vertebrate Genomes project, which is aiming to get very high quality reference genome assemblies for all vertebrate species. This project has been going on for a little while now, and we're in phase 1, where we're creating good genome assemblies for one species per vertebrate order, which is about 260 species or so. And for this first phase, we're focused, we've sort of thrown the kitchen sink at the problem in terms of technologies. So we're, for each ordinal genome, we're doing PacBio, 10x, BioNano, and HiFi as well. In addition, sort of around the same time as the Vertebrate Genomes project was sort of kicking off, Sanger was celebrating its 25th anniversary, and there was this project started to sequence 25 genomes from Britain, representing local biodiversity, and that has also led into a larger project which was announced fairly recently, called the Darwin Tree of Life project, which is a project to sequence all eukaryotic UK species in the coming years, and that's a collaboration between a number of centres here in the UK. So just to give you an idea of what we're targeting for the Vertebrate Genomes project, at least, is assemblies which meet this G10K quality metric, which is basically contig N50 greater than a megabase, scaffold N50 greater than 10 megabases, and at least 90% of the genome assigned to chromosomes. And then, importantly, also getting base call quality above QV40 and trying to, as much as possible, get diploid haplotypes out of the assembly process. So, so we're sort of in the phase of trying to scale up these assemblies if we're going to meet the goals of these projects. So in that context, we're evaluating the 8M system, and we ran a number of different animal samples, non-humans mostly, and I'll just run through some of the samples that we're gonna talk about here. So the first off was the brown trout. This is a nice species that we got from a collaborator in Norway, and it is actually a double haploid sample, which is quite important here because the brown trout genome has a unresolved whole genome duplication, which means some of the genome is in a tetraploid state still. So we previously had CLR data, and we have the other data types from the VGP project for this. We've actually gone through and curated and released an assembly, but we were curious what the CCS data was going to look like for this one. So that's first off. The ploughfish, this is one where we had previously had a CLR assembly, which proved quite difficult. Some fish assemblies, especially that we're coming across in the Vertebrate Genomes project, proving more difficult than a lot of the other species types like birds and etc. So we were curious how CCS was going to perform there. For this Sicilian, which is a large amphibian genome, we were curious how few smart cells we would be able to get to get a good assembly from this. The hawk moth is one which was done for the 25 genomes, and we had existing PacBio data from before and after the switch from version 2 to version 3 chemistry, and so we're curious how CCS would change things there. And for the sterlet, this is another interesting fish where we have an unresolved whole genome duplication, and in this case we have access to a trio where we can do trio binning to separate out the haplotypes. The milkfish is one where we decided, given a lot of the fish in the Vertebrate Genomes project are proving quite difficult, this looked like a sort of normal, typical fish. We thought we'd, from the same starting material, produce both CCS and CLR data and put it out there as a data set for comparisons. And then lastly, another amphibian where we were trying, it's a large genome, trying to get see how few smart cells we could do to get a good quality genome assembly out of this. So this is the rundown of our yields, etc., from the various runs. You notice it's not 32 smart cells here. That's because the leads projects that I focused on were sort of half of what we ran here at Sanger. The other half was run as part of the cancer program, so mainly on human cancer samples. But you can see our yields here we were quite happy with them. We had one failure in the HiFi run here, but then that subsequently ran and gave a good yield here. And for the CLR runs, we did started off a little rocky, but then we've kind of got more consistent towards the end, and we've had runs since this, which have been in the high levels here, sort of 80 to 90 to 100 plus gigs. So we're quite happy that after some initial teething, it seems to be running quite consistently in our hands. So I was just going to run through two of these samples here. So I'll run through some results from the sterlet, where we have the trio assembly, and from the milkfish, just to give you an idea of some of the qualities of the data. So for the sterlet, we sequenced 2 smart cells. Both had over 100 gigs in yield per smart cell. It's about a 1.7 gig genome, which means we're quite high coverage in CLR data here, and the read N50s were quite good as well. So as I said, we have a trio here, and we've sequenced the trio, the parents, to about 40x on an Illumina HiSeq-X, which means we can apply this nice method from Adam Phillippe's group, which is to use the k-mers in the parental Illumina data to bin the PacBio reads into those which came from the paternal haplotype and those which came from the maternal haplotype. So you can see in the distributions here on the right of the k-mer distributions, this first peak here is the things which are single copy in the one of the parents, so that means those are the hets essentially. The second peak is the things which are 2 copy, so it's the diploid parts of the genome, and this peak over to the right are the things which are 4 copy, so the tetraploid parts of the genome. So using those k-mers unique to either the paternal or the maternal Illumina data, we can assign the PacBio reads, and that worked quite well here. So after trio binning, we had essentially the same amount of data assigned to both the maternal and paternal haplotypes. In the bottom left here is the read distribution. In blue is the read distribution for all the data, and then stacked pretty much on top of each other are the maternal and paternal read distribution. And then here on the right, you can see this is inverse cumulative. So again, blue is all the data, and right on top of each other are the maternal and paternal, and this sort of says we have about 50x coverage at reads of 20 kb or greater. So the assemblies from, so assembling each of the parentally binned PacBio data separately, we get quite nice assemblies here. So contig N50 around 6 or 7 megabases. The genome size looks quite accurate, quite spot on. This code here, so if for those not familiar, this is a Busco scores. So this is looking for single copy orthologous genes, which should be a single copy in a particular lineage. So C here is finding if you found those genes and they're complete. And then that's split out into single they're complete and single copy in your assembly, or complete but duplicated in your assembly. and then F is fragmented and M is missing. So for fish genomes, this 92% is quite good, especially at this stage of an assembly, pre-Illumina polishing or anything like that. And then I guess the interesting thing here is the duplication rate is quite high, and that's because of the tetraploidy. We have a lot of multi-copy genes in this genome. And this plot sort of shows that we are separating out the haplotypes as we expect. So here, each blob represents a contig, and the size of the blob is how many k-mers are in that contig. So it's roughly the size of the contig, proportional to the size of the contig. And then its position on the x or the y-axis is how many of those k-mers are assigned to either paternal or the maternal or paternal unique k-mers. And for a lot of assemblies, you'd get things off the axes here, but for these, we've separated them out quite nicely. Arrow gives quite a similar assembly, slightly lower contig N50. It's still quite good from the looks of it. Watchtower here, again, it looks okay, but the genome size for Watchtower here is quite small. So I think you'll see why in the next slide. So here we're doing a CAP spectra plot. So again, this is the k-mer distribution of the, in this case, the 10x reads from the child, from the individual we're assembling. And so things on the left there are single copy, things in the middle peak are things which are 2 copy, and things on the right are 4 copy. And you can see they're color coded by if in the assembly they are actually seen as what copy they're seen in the actual assembly. So ideally we have red here at the single copy peak, all purple here for 2 times at the 2 copy peak, and over here 4 times at the 4 copy peak. And we've roughly seen that here in the Falcon assembly, with some of this green stuff here at the top being stuff which is 3 copy where it should be 2 copy. But that's, I'll come to that in a little bit. Canoe assembly looks quite similar. The Watchtower assembly, you can see what's happened here is that there's a lot of stuff which is, all this black stuff at the bottom is stuff which is essentially 0 copy. It's not seen in the assembly for Watchtower, so it's collapsed it out. And this red stuff here under the what should be all purple are things where it should be 2 copy, but it's 1 copy, so it's being collapsed in the Watchtower assembly. So here the duplication in the Falcon and Canoe is actually due to overlapping contigs, which we can post-process and clean up a bit now. And the Watchtower assembly in this case is collapsing things overly. We're continuing with this assembly. So for the milkfish, we, as I said, we generated CLR and CCS data. So the CCS, this is just what we got for the CCS data. So we ended up with 26x coverage in CCS reads, which was quite good. Mean about 10K, which is expected, and the Q score of a median of about 31. So we assembled with Watchtower from this. So we've got an OK contig N50, 1.6 meg. And I guess the nice thing about this is you don't have to polish and you still get a good Busker completeness score here of 95%. So Jason Chin got in contact when I put this data out. He has a new, so Jason Chin wrote Falcon initially, and he has a new assembler, Peregrine, which works on HiFi data. He took that data and tried it out. So he has made an assembly which is looks slightly better in terms of stats than the Watchtower assembly here. So for this data, we have the CLR data set as well, and that's sort of gone through our normal BGP pipeline. The Falcon assembly is sort of roughly 2.5 meg contig N50, just doing Falcon unzip, and then we've pushed it through the rest of our pipeline where we've scaffolded and curated and our current assembly is now 3.6 meg contig and 50 in 25 scaffolds at scaffold and 50 or 54 meg. Most of it is assigned to 16 chromosomes, and we're quite happy with this. Though we have another version in the pipeline which has a lot higher contiguity, and we're probably going to reconsider this other one soon. So yeah, this is quite a nice test set for people if they want to check out the quality of CCS data versus the CLR assembly. So the data is available. So VGP is putting out all its data as it's generated. There's a data use policy you can check out here. If you go to vgp.github.io, you can see all the, not just this data, but all the data that the VGP is generating. This here is a summary of where you can get the of the samples that we've run here on the Sequel II that are VGP and can be released in this manner. So yeah, we're quite happy with the Sequel IIs so far. It's a huge leap for these in scaling and affordability for these tree of life genome assembly projects that we're working on. The yields are great, and the vertebrate assemblies are looking quite good. From now we can do them from a single or a handful of smart cells. We have generated some good quality CCS data, but they were still evaluating exactly how those assemblies, how to best use the CCS assemblies for non-human assembly at the moment. So with that, I'll finish, and I want to highlight especially Craig, Karen, and Michelle, who are the people in the lab running the machine and doing extractions and the libraries and everything. And the vertebrate genomes as well. So yeah, thanks for your time, and I guess we're going to head to questions in a sec.
Great. Thank you very much, Shane, and thanks to all the speakers. And I want to, before we get to Q&As, I want to point out that, and Luke mentioned this in one of the slides, that although the system has only been formally at customer and researcher site for less than 2 months, we already have 3 sites of certified service providers. So if you have a project that you want to run on the Sequel II system, Hudson Alpha Genome Sequencing Center, the ICANN Institute of Data Science and Genomic Technology in New York City, and Luke's institution and facility at the University of Maryland School of Medicine, the Institute for Genome Sciences, are open for projects, and so please feel free to contact them. So with that, we'll go to questions. The first question maybe is one for me about also about data access. Shane mentioned that. So the question is, do you have comparisons of non-HiFi assemblies versus HiFi assemblies? And can I access the subread data for the HDSV samples? So with regard to, those are related questions. And in anticipation for this, the data sets are available for you to explore and to test. So in the recording, you'll have a screenshot of this with the links and so forth. But we've made quite a number of data sets now available from the Sequel II system. With regard to comparisons of non-HiFi, so CLR assemblies versus HiFi assemblies, there are 2 manuscripts in the bioarchives. One, a collaboration of our researchers with, I believe, 14 or 17 other institutions comparing human genome, CLR versus HiFi, and then more recently, Evan Eichler and researchers at PacBio collaborated to look at a CHM13 CLR CCS dataset, and so I invite you to explore those and look those up in the bioarchives. There is a question for Kiran, I believe, asking about what's the sensitivity of calling rare variants. Can you find heterozygous or heterogeneous variants, or those rare molecules with unique somatic mutation signatures get to be filtered out because it doesn't follow the germline segregation?
Right, so we have the same question. The short answer to that question is I don't know yet. The longer answer to that question is this is precisely why our initial run of the instrument was to collect trio samples. Because with the trios, we can use the Mendelian concordance to establish a ground truth data set, and then go back and then run a battery of short and large variant callers on these data sets in order to answer that question. That's precisely why we're going to contribute these data sets to HGSV, so that we can start taking a look at all of the sensitivity and specificity metrics across a wide number of callers.
Great, thank you. Next question is for Luke. Do you have any recommendation to get rid of the human genomic DNA for shotgun metagenomes?
Unfortunately, not any great suggestions. I mean, there are, relatively limited ways to remove it at the nucleic acid level, but most of the time, if you're doing shotgun metagenomes, it's removing it at the data level once you've generated the data, and there's a number of tools available out there that are pretty well publicized for filtering the data.
Great. The next question, I guess, maybe is for me again. How is the Sequel II performance in terms of transcript calling, especially the ones with different 5' or 3' ends? And so while we covered a large breadth of application areas, human, microbial, metagenomics, and animals and plants, we didn't have, there's so many application areas that you can't fit them in an hour. So IsoSeq, unfortunately, was only mentioned briefly, I think by Luke, but IsoSeq works very well on Sequel II as well as it did on Sequel I, but the added benefit, of course, is that you now get a much higher number of reads, and the HiFi read, the CCS principle, of course, is very advantageous for differentiating and segregating even closely related isoforms, seeing the different 3' and 5' ends, looking at UTR, variability and so forth. So IsoSeq is and remains, I think, one of our fastest growing application areas, and is very suitable in, of course, a complementary mode together with short-read RNA-seq, where you get a lot of counts from RNA-seq, and then you get the information about transcript structure with increasing depth now at the Sequel with the Sequel II system for the most comprehensive characterization of a sample's transcriptome. There's a question for Shane. So there, the question is, you pointed that for many of these organisms, you have many different kinds of technologies, and so the question is, do you need all of them, and if so, what's the relative contributions of the different technologies that you apply to these samples?
Yeah, it's a good question. That's one of the aims of this first phase of the vertebrate genomes project, is to try and find out which data types are the best sort of bang for the buck in terms of working best across a large range of species. So at the moment, so I guess at the moment we're using 10x, BioNano, and HiC all for scaffolding, and they work on different ranges. So 10x tends to work quite well at doing getting good quality scaffolds on a short range, so on the length that you have molecules for the data. And then BioNano tends to give you sort of a middle range, and then HiC and a particularly long range. And then of the three, the BioNano tends to be the most robust in terms of the most reliable in terms of having the scaffolds that it makes often quite reliable. HiC, you can still run into certain issues depending on your HiC data, but it's sort of, we're still sort of trying to work it out the best. And the other thing is we apply them sequentially at the moment, which is not ideal. Like ideally there will be methods that we can combine all the data types at once and choose the best way to scaffold. With HiC, of course, there's other methods out there at the moment, Falcon Phase, where you can use it to phase your assembly as well, which is something we're trying to look at the moment.
Yeah, so but they all start with the PacBio assembly, and then you put the scaffolding technologies on top of those.
Exactly. Yeah. We found that the 10x only assemblies haven't been great in our hands, haven't met the metrics that we're trying to get for the BGP.
Good.
And for the.
Yeah, thank you, Shane. And maybe in view of the time, the last question, and maybe that's one for me again, what is the error rate in Sequel II data as compared to Sequel data? And so we've actually improved upon that. So you need less passes to get to a certain accuracy in the HiFi mode, in the CCS mode. So depending on the threshold that you would like to set, in Sequel, it took about 4 passes to get to Q20 or 99%, and that's reduced to 3 passes. And for Q30 or 99.9% accurate CCS reads, it previously took about 10 passes on Sequel, and now that's reduced to 8 passes. So we've actually improved on the raw read accuracy that gives you faster consensus and less coverage is needed to get to a certain accurate read quality. So with that, in view of the time, in closing, I'd like to thank Kiran, Luke, and Shane for their time today and for the terrific presentations they shared. As I already mentioned, we have recorded this webinar and we'll have it available for download in the next several days. So keep an eye out for a follow-up email with a link to the recording. And in closing, I want to briefly mention 3 things. On the slide, you see the certified service providers that already provide Sequel II service. You can always stay up to date on upcoming webinars as well as conferences, trade shows. You can meet with us at visiting and bookmarking pacb.com/events. And as Luke mentioned in his presentation, if you have an interesting research project that could benefit from smart sequencing, also consider applying for our Metagenomics Smart Grant and win free sequencing. For more information, visit pacb.com/smartgrant. So thanks again for all joining us today. I hope you'll join us again on a future webinar. And with that, take care and have a wonderful day.